noticed it. These little tips are starting to... All right. So when we look at the idea of mitosis, I just want to make sure I go over this one last time with you, and we're all clear. We're looking at a cell. I usually draw a cell round, but a cell has different shapes. Some cells, like epithelial cells, can look like so, where they have their... they have all the little structures that they should have, and they're really tightly connected to one another. And they're skin cells, right? They create a smooth surface that protects you, the inside of your body from the outside, right? And there's layers to the dermis. There's an epidermis, there's a mesoderm, there's an endoderm, etc. And there's actually very numerous layers to the skin. It's an organ. It's the largest organ of your body. It's incredibly important. When you burn your skin, especially if it's a large portion of your skin, it could actually kill you, even though you're not diseased. It's very difficult to survive large areas of your body's skin being damaged. So in your intestines, you have different cell shapes. They have these little microvilli, and they are also tightly held together because they also have to protect you from the bacteria that are in your intestine. And they're specialized in, they became different, they became specialized, they became differentiated so that they can increase the amount of surface area so that they can absorb, right? That's what intestinal cells do. Your lungs also do that. Your lung cells also have little microvilli, and all these that are designed to absorb material, whether they be absorbing oxygen in your lungs or absorbing food in your intestines. They maximize their surface area, and they have little capillaries, tiny blood vessels that come near them on the other end that allow for that exchange. And of course, this is your blood, and it's in this blood. This is where all that material is transferred to as it goes to your liver or to your wherever it's going. So and of course, the blood vessels are made of cells, right? They're made of cells that all this has to do with that osmosis and endocytosis and all this stuff that we've talked about up to this point. The blood vessels are made of cells that are all connected. All of them had gone through mitosis, divided and organized into shapes, and every cell has a different set of genes that are turned on. Even though they all have the same DNA, I keep saying that over and over again, they all have different functions, so they all have different genes that are turned on, they all have different shapes, and in order to do those functions, remember, shape and structure and function are intricately linked. Muscle cells are striated. They tend to be. And why would a muscle cell be this way? It would have this shape, and so it can pull. Because what do muscle cells do? They pull. By the way, that's all your muscles do, is they pull. They never push. They just pull in opposite directions. So if you have a bicep, you have a tricep. And a bicep pulls in one direction, and a tricep pulls in the other direction. So you're always pulling. And so they're cables. They're made of these muscle fibers that are just pull. They're connected from one end to the other by ligaments and tendons, and they gives them an anchor, and the fibers pull each other. Of course, the structure is they tend to be long and striated, and I guess, of course, they have the receptors that allow for ion exchange and all the stuff that we covered, like the action potential from a neuron, et cetera, et cetera. Hair cells. We looked at the nerve types. They're called hair cells, but you also have cells in your scalp that produce hair, right? They start producing it. They just make hair. Hair is amino acids that are all intertwined, wound up around each other, and the cells just sit there. Your follicles, they're called follicles. They're hairs, cells in your follicle. They're just producing protein. That's all they do is all day. You have glandular cells, little oblong cells in your liver and in your pancreas that whose job it is just to produce hormones. And that's all they do all day is they produce hormones. Again, they're going to have blood vessels that go near them made of cells. So you're all made, everything that you know of is made of cells. Your eye has a bunch of, the oddly shaped, things that are like rods and cones, right? You've heard of rods and cones that in the back of your eye called the retina. But then you have cells here in the beginning that form a layer that produce this layer called the cornea. And you have muscles here on each side, little tiny muscle cells that change the shape of your eye. And that squint your eye closed, and you have the iris that can open and close. And there's pigment cells, there's melanocytes in the eye as well. And the fluid, there's a fluid in here as well. So this organ called the eye is made of numerous tissues of many different kinds of cells. Each one has its own set of DNA that are turned on and off. So every organ you can look at and dissect is called histology, and you can look at the different cells and tissues that make up these organs. And it all started from that one single fertilized egg, that zygote that had all the information. It's amazing to me. All the information that you needed to become all these different cell types. As they differentiated, as they became different. So let's take a look at mitosis specifically. We have this thing called DNA. It's double-stranded, as we've talked about a lot. It's 5' to 3' on one side, and it's 3' to 5' on the other. They're anti-parallel. Along this really long, billions of nucleotides. I want to remind you what a nucleotide is. A nucleotide is when you have a ribose sugar, and it has on one end some kind of nitrogenous base, and on the other you have a phosphate attached, and on this end is connected to another phosphate. So you have this sugar phosphate backbone, and then you have this hydrogen bonding between the nucleotide, the nitrogenous, the two chains, the double-stranded DNA. When you take that, when it's unwound, it's called euchromatin or chromatin. It's all just unwound. It has to be unwound and open so that it can be read. And we'll talk about how we do transcription later, but you have to be able to read the message. The message is here. There's the message right along the inside. These nitrogenous bases, you have to read them. We have a protein, an enzyme complex that'll literally read them. ATGC, ATTC, and depending on what they feel on one end, when I say read, I mean feel. It's a chemical reaction. They feel on one end. On the other, they put that amino acid together. So that literally tells you what protein you need to make on that code. We call those genes. Genes. So a gene is an area on this area here, for instance, that's a bad color. This area here, for instance, could be a gene. So this here, this little boxed out area could be a gene. It could be a gene for anything. It's a protein that does something. What does it do? Well, it depends on the gene. Maybe it combines with some other genes that make your skin darker. Maybe it makes your skin light. Maybe it helps control skin tone. Maybe it helps control how much hair you have. I just read an article just yesterday that we found that humans have the gene to have hair all over our body, like our ancestors did, right? Like we have the genes that if we turned them on, they would make us hairy like apes. Isn't that interesting? That's really, I was like, this is another more cooperation that evolution is likely a good explanation of what's going on. Because if we turn these genes on, we would have hair all over our bodies. The point is that there's these genes, and it's not just the relationship we're going to study in the next few weeks, all the different combinations of how these genes work, but they produce a protein that does something. The protein does something, or the RNA does something. Yes?
This may be an off-topic question, but do we know how exactly the eye evolved?
Yeah, we do have a lot of evidence to how the eye evolved. For instance, just a quick suggestion to look up photosensitive spots on single-celled organisms. Like that's the simplest eye, right? We know that there's not single cell, maybe they're microscopic organisms. They have small areas on the back of these small microscopic organisms that are sensitive to light. And then you could see all the little different bits that if you put them together over millions of years, you would expect to see an eye. And there's all kinds of evidence like where naked mole rats have lost their eyes. And you can see how they have eye sockets but no eyes, they're blind. But why would you have the structures there if you're blind and you live your whole life underground? And you can go and look up all kinds of different lines of evidence that show clearly that how we evolved. And we could take apart not just the DNA of the eye, but also each individual structure, and we can identify them in other creatures. But we're not going to do that today because we don't have time. So these genes, they control something. And you learned in middle school, I believe, that this idea of big A and little a. Is that correct? Yeah. Right. Dominant recessive. It's more complicated than that, but we're going to go ahead and just go with that for now. Big A or a little a. All that means is that there's two versions of that gene, right? It's like you could get one from mom and the other one from your dad, or maybe you got both the same versions from mom. Maybe your mom and dad had the same version of that gene. One version overwhelms the other, does a better job at what it does, whatever that is. If it's making pigment, it makes a better pigment. And so we call that dominant. It shows up. Dominant just means it shows up. So just get that in your head now, that if you see this word dominant, it just means shows up. And recessive just means you don't see it unless you have two of the recessives. Huh? You have two versions of every gene, right? You got one from mom and one from dad. Because we'll go ahead. So you got recessive. You only see the recessive, which is over here. You only see that one if you have two versions of them. You have to have two versions to see it. That's the two copies of recessive. So let's say one gene, big A, this is really simple. It doesn't work this simply in humans for most genes. Mendel was very lucky that he used P's because the genetics is much simpler than the reality of most other genes, than a lot of our genes in humans. But let's say it's simple. Let's say it's a plant or something. And you see it's a big A. Big A makes it tall. If you have two little A's, it's short. So if you have a big A and a little a, what is it? Tall. No, yours is a different kind of genetics, not dominant recessive. If it's dominant recessive, if you have the dominant, that's the one you see. In recessive, you don't see it. The trait is what you see. The genetics is the big A or the little a, the version of the gene. Is that clear? That medium thing is what we call incomplete dominance. Right? Incomplete dominance, and there's another one, or codominance. In that case, it would be incomplete dominance. Yeah.
So is there really a difference between like 2 dominant and 1 dominant and 1 recessive?
Very good question. In a true dominant recessive inheritance, no, there isn't. In the trait that you see, right? Only for dominant recessive, I want to make that clear, because what Georgia said is absolutely right. Some of the gene inheritance is going to be you get a medium. In fact, that's how height in humans works, by the way. That's why 2 short people and how tall someone is actually controlled by multiple genes. And you could have 2 tall people having a short person child because it's complicated. It's the mixture of all these different genes that you've gotten from both parents that determines how tall you are. Same with skin tone. There's not black or white. There's 64 different skin tones, all controlled by 8 different genes with multiple alleles in each gene. Like I'm just looking at A, big A or little a. But in blood types, we've already learned there's A, B, O. So there's 3 alleles in ABO blood groups. That's just 3. Some of these have multiple alleles. Now things get complicated, right? But to keep it simple, dominant means you see it. Recessive, you need 2 of them to see it. Is that okay? All right. So when we look at, I'm going to use humans because I think we're all interested in humans. We have chromosomes that are wound up. This is how it is when we're using the DNA. When we're not using it, we're ready to, after we're copied it, we pack it up. Why are we packing it up? Because we got to move it. Mitosis means dividing the DNA. You don't just, if your mom said you're moving tomorrow, you're not just going to go and grab a bunch of clothes and move out the door and throw it in the car, right? I mean, you might, but I promise you'd lose half your socks for sure, right? That's gone. You'll never have a pair of socks that match again. And then T-shirts, you're going to forget your best T-shirt, you're going to forget an Xbox controller, you're going to forget, you're going to lose half your stuff. It's not going to be organized. When you get onto the other place, you'll be lucky if you have exactly everything it doesn't work that way. That's why you get boxes and you pack things up and you label the box and you make sure you got all your stuff. And then when you move stuff professionally, they just label it and say kitchen, boys' room, girls' room, mom and dad's room, that kind of stuff. Why do you do that? Because it makes sense. It's the best way to do it and keep everything where it's supposed to be. Your body does the same thing. Life does the same thing. When you're ready to divide the DNA, not only do you have to copy it, so you have to unwind it. It has to be unwound so you can copy it because you can't read it. If it's not unwound, you can't copy it if you can't read it. Does that make sense? So it's all unwound at first. You make copies, and very carefully. So if this is what a chromosome normally looks like, after you've done S phase, your chromosome looks like this. It's two copies that are connected together by a centromere. Are we good? And it's that little structure right here. In the middle of that is something called a kinetic core. It's going to be important in a little bit when we get to anaphase. So when these 2 things here, they're 2 copies of the DNA. They're copies of the DNA. So let me make this. Let me be sexist and make this dad's chromosome and make it blue. Or genderist, not sexist, genderist. Let's say this is dad's chromosome, it's blue. Okay? There's 2 copies here. You only got one of these copies. This is chromosome number 1. Maybe this chromosome has the genes for height and a gene for skin tone and a gene for your right brain development or your eye color or your eye shape or the shape of your nose or the gene for lung or for genes that are involved in liver, genes that are involved in your brain. I don't know. But that's the genes that's on this chromosome number 1. I'm making it up. Now you have 2 copies of those genes. They're both chromosome number 1. They both came from dad because they're copied. You copied it during S phase. So this is dad's chromosome number 1. We don't call these both chromosomes because this is all one chromosome because they're attached. It's one thing. But we do call them sister chromatids. Now, sister chromatids are just copies of the same chromosome. They're just connected together. Now, you have 2 chromosome number 1s, don't you? You got one from mom and then one from dad, and what's the other one? One from mom. They have the same genes. They still have the centrosome. They still have a kinetic core. All right, they're still called sister chromatids. These two are sister chromatids. But these two are the same chromosome with same genes, but we call them homologs. Why? Because they're both chromosome number 1. They have the same genes. What's the difference, though? Why do we even care that one came from mom and one came from dad if they have the same genes? Close. They have the same genes. What do they have that's different? That's a dominant recessive. That's good. They're different alleles. We call those versions. The versions, big A, little a, we call them alleles. Yeah.
What's the role of the centrosome in this case?
It's holding them together. Keep them together. Like I said, when you got socks, you're done washing your socks. What do you do with your socks? What do you do with your? But you don't just throw them in a container. What do you do with unicorn socks, or your pink socks versus the red socks versus the white? You put them, you match them, and you somehow affix them to each other, right? In the case of socks, you kind of fold them into each other. Well, this centromere is doing the same thing. You made a copy. You got 2 of them. You want to keep these together. You stick them together with a little tape, little Velcro. This protein complex is doing just that. It's just holding these together. So we call these homologs. Now, I'm going to use a different color. I use gray, I get. No. Light blue. All right. So here, maybe this one here, they're the same. So maybe they have little a. The allele, the versions of the gene are called alleles. All right, they're called alleles. So you have a gene, and I don't know what little a does. Who knows? How tall you are, how dark you are, how whatever. The version is little a. Mom might have a version that's big A. It's the same gene. It's just a different version. Maybe instead of a G, they have a T there at a specific site. Maybe instead of a C, they have a G. Maybe they have instead of an A, they have a C. Whatever. And that changes the protein enough that one becomes dominant over the other. Are we clear on that? Yeah.
Is this allele variation the only reason why we're distinct from each other?
Yes, it's one of the main reasons we're distinct. That and the fact, that would be the only reason humans are distinct from each other. So, what if then dad had, a big D here, and mom had also a big D? See, they could have the same alleles. They could be different. But there's 10,000 genes at least 10,000 genes in the human genome. So you can see how different the combination you can get. So there's your 2 chromosomes. Now that's what you have, and I remember you have to do 23 of these pairs, or a total of 46 of these chromosomes in humans, right? 46 chromosomes in humans are 23 pairs. So you gotta keep this. That's why it's so important to keep it organized. In some creatures, plants, I think it's corn, you can get up to 100 plus chromosomes. That's before you do duplication. Remember, you've duplicated this, so it's not 46. What is it now after you've duplicated? How many copies of the DNA do you have? 92. You still only have 46 chromosomes, though, because they're connected, but you have 92 copies. Does that make sense to everybody?
Yeah.
Okay.
And is that where the extra chromosome for
No, we will talk about it. Down syndrome extra chromosome is what we call non-disjunction, which means when you're separating them, instead of getting one going to each cell, during meiosis, you accidentally keep 2 in one cell, and then when the sperm meets the egg, now you got 3. All right, and that's what we'd call trisomy. That's just one example. Trisomy because you have 3 copies. In any case, that's later. That's for meiosis. Right now, I just want to make sure we're all clear on mitosis. So that's what you're going to have the lab on Thursday. Yeah. So again, looking at the cell cycle, remembering that you start off with one cell. You start off with one cell, and then you go into G1, and then you go into S phase. By the way, at the end of G1, before you go into S phase, you have the most important regulatory, what we call checkpoint. Checkpoint in the cell cycle. If there's something wrong, and how does your cell know if there's something wrong? It knows there's something wrong because it's all about the concentration of chemicals and structures in a particular area. If your concentration's off, your cell's going to know it. So if at that checkpoint, so 2 options, you got 2 options. Is the concentration of the signal, whatever the signal molecule is, there's different signals for different cells. Is it concentrated okay? Is it right? If the answer is, right, the concentration of the signal okay. If yes, then continue. Continue the cell cycle. That's it. Just keeps going. If it's no, you have 2 options. Option 1, fix the problem. Option 2, die. So it's called apoptosis. This is good. Cell death is normal. It's expected. It's healthy. It's part of development. Death is part of development. So let's be clear on that. I'll improve on that as we move along. Apoptosis is important. If there is something wrong and you cannot fix it and it doesn't die, we call that cancer. That's bad. So cell death is preferable. Yeah.
Is apoptosis just cell death?
Cell death, that's it. That's a good question. Apoptosis just means cell death. Yeah.
So when the function of apoptosis isn't working,
That's when, because it doesn't work and it keeps going. The checkpoint's ignored and it keeps doing cell cycle over and over and over and over, and nothing's going to stop it because it's not going to die on its own. Now, most of the time when this happens, your body has all kinds of checkpoints. Otherwise, we wouldn't live to be 100 or 90 or 80 or 60 or whatever. We die at 1. Because you need this is many divisions, right? Many cells. If this continues, usually your body will pick up the cancer, ladies, and kill it. Your white blood cells will destroy it. There's something wrong with the cell signals that tell the cell, this is us. Cell's like, uh-oh. But sometimes, a lot of times with cancer, because they're your cells, your body can't tell the difference between the cancer cell and the normal cell. So it looks like a brain. I mean, it doesn't know that you have brain tissue in your stomach. How does it know that? It doesn't. It's just either it's you or it's not you. Yeah.
What are some things that would cause the checkpoint to get ignored?
That's a mutation. So usually it's a mutation. Sometimes it's a chemical. We call it carcinogen, right? That causes a mutation. in the proteins, that in the signaling molecule. There's all kinds of complicated, we're not gonna get into details of how these are regulated. In AP we are, but we have these things called cyclins and CDKs, cyclin-dependent kinases, and there's all kinds of hormones that signaling molecules that encourage the cell cycle and those that repress the cell cycle. So for instance, there's tumor repressor genes, and there's genes that are growth factors that actually encourage the cell cycle. You need both, right? You need to be able to turn the growth cycle, the cell cycle on and turn it off. So you have all these signaling molecules we're not going to get into in this class. You don't got to worry about it, but just know that it's there. Yeah.
So a mutation would cause the cell to miss the checkpoint?
That's right. The mutation could maybe change the signal so that the checkpoint thinks it's okay. Maybe the mutation messes with the receptor. So who knows? There's different mutations for each one. We know there's a mutation. We know a specific mutation, for instance, that has to do with breast cancer. We know what that mutation is. In fact, you know Angelina Jolie inherited that gene from her mother, and she had it's an aggressive gene. Aggressive form of this cancer is linked to this gene. She had 2 copies, so she had both her breasts removed because she did not want to go through what her mother went through. So this is the reality of these checkpoints. So it goes on, S phase, you make the copies. On the other end, oops. Yeah.
So like the signal, does like a certain process happen, or is it just like a quick signal?
It's a good question. It's a process. It's complicated. It's a path. I don't want to make you think like it's impossible to understand. It is possible to understand it, okay? The issue is that it's beyond the scope of this class, okay? If you want to learn about it later, we can talk about it. But so M phase is the part we're talking about now, which is mitosis happens here. In M phase, you have 2 things going on: mitosis, the division of the nucleus, and then you have cytokinesis, the division of the cell. So let's be clear on that. And then, of course, you end up with 2 cells, which then can go through the cell cycle themselves again and do it over and over again. You have another checkpoint here. There's another checkpoint in G2, and there's a checkpoint here. So there's 3 checkpoints. This number 1 is probably the biggest regulatory checkpoint, but they all check for different things. But each checkpoint is made specifically. Is there something wrong? No? Continue. Yes? Fix. Can't fix? Die. That's it. Yeah.
Is it a lot of codominance?
It's a complicated situation. Is it time to go? Is that why people are packing? All right. Well, I'll try to finish this today and post the video in case you need help for Thursday. For Wednesday.